Welcome to the latest podcast from the Plastic Surgery Journal Club. Each month we review and appraise a journal article from PRS and summarize it for you in this short podcast. The full paper can be obtained from the PRS Journal website.
Hi, and welcome to the June 2016 Plastic Surgery Journal Club podcast, coming to you from Sydney, Australia. My name is Dr. Damien Marucci, and I'm with Dr. Mark Rahman, who is a plastic surgery registrar. So, Mark, the first paper that we did tonight was Bacterial Biofilm Infection Detected in Breast Implant Associated Anaplastic Large Cell Lymphoma. This is from the June 2016 PRS, and it's by a multinational group, but headed by Professor Anand here in Sydney. So what was this paper about in a nutshell?
So essentially this paper was building on previous work that this group has done. They have previously implicated biofilm in capsule formation around implants and also in ALCL. And the question they wanted to ask here was, do the bacterial microbiomes differ in non-tumour capsules from those with ALCL capsules?
So basically they got a whole lot of ALCL samples from around the world, and they compared them to capsular contracture samples in non-tumour patients. How are they able to detect microorganisms or biofilms in these samples?
So they used a variety of methods to detect the presence of biofilm. They used a real-time polymerase chain reaction, next generation sequencing, FISH, and scanning electron microscopy. They looked at 19 specimens of ALCL, and they compared these to 12 non-tumour specimens. Interestingly, they also looked at 3 specimens from the contralateral breasts of ALCL patients, so non-ALCL specimens, but from the same patients.
And what were their results?
Their results were they found that very high bacterial counts in both non-tumour capsules and ALCL capsules, as we would expect. But most interestingly, they found a very high proportion of a specific bacterial species in the ALCL group, specifically Ralstonia species, and they found that the vast majority of the microbiome was made up of these species in ALCL capsules. Interestingly, they also found that the same species was present in the contralateral non-ALCL capsules,
but in a lower number.
But in a lower number.
Yeah. So they sort of came up with the conclusion that if someone, or they hypothesized, if someone had a gram-positive biofilm, it tend to lead to capsular contracture. A gram-negative one leads to lymphocyte activation and potentially leading on as a causation of ALCL. So what did people think about this paper?
Yeah, I think the reaction to the paper was very positive. It's obviously a tremendous amount of work's gone into this, and really ALCL is a very rare and up until now pretty poorly understood condition, or I should say implant-related ALCL is. And this paper really seems to have moved our understanding forward quite dramatically.
One of the issues is obviously, as you brought up in the discussion, correlation does not prove causation, but certainly this is a pretty heavy smoking gun, and there is quite a marked association with this very unusual Ralstonia species and ALCL in the specimens that they've looked at, as opposed to ordinary capsular contracture specimens.
Absolutely, and I think as you pointed out as well, that one would have to question how you could feasibly prove causation in an ethical way.
Yeah, whether you could have an animal model, because I know obviously Anandiva's group has inoculated pig implants with human staph epidermidis, and I don't know whether his group is looking at maybe inoculating with Ralstonia species or the like. But it was a very good paper, and it was a good discussion.
Excellent.
Thank you.
Thank you for listening. For more of our podcasts, head to soundcloud.com or subscribe to us on iTunes and search Plastic Surgery Journals. Remember to like us on Facebook and follow us on Twitter. Thanks also to the PRS Journal team for their ongoing support.